Good afternoon, everyone. I know we usually start at MIT time, 5 minutes after 3, but we want to make sure that Professor Arnold makes a plane. And we're going to get started at 3 with the introduction that I usually give for the Hottel lecture while people continue to come in and make yourselves at home, find a seat while we do the introduction. So it's my great pleasure to welcome you to the 2021 Hoyt C. Hottel Lectureship. Before I introduce our incredible speaker, I'd like to tell you a little bit about Professor Hoyt C. Hottel, for whom this lectureship is named. Professor Hoyt came to MIT from Indiana in 1922 at the age of 19, having just gotten his AB, or Bachelor of Arts, in Chemistry from Indiana University. And he went on to get his Master's in Chemical Engineering in 1924 and became an assistant professor at MIT immediately afterward, after doing an assistant directorship at the practice school from 1924 to 1925. He was a professor in our department from 1928, when he was the assistant professor of fuel and gas engineering, to 1968, when he became professor emeritus, and during that time held the first Carbon P. Dove's Professor of Chemical Engineering chair. He remained active in the department until his death in 1998. That's 75 years of dedication to the department. Now, Professor Hottel will be remembered for his intensity, his intellect, and his integrity. His lectures tended to make students think, and he was very active in seminal contributions to a number of areas in energy, including the measurement of gas emissivity. He established the mathematical framework for the quantitative treatment of furnaces and the first systematic investigation of the laminar furnaces. And he also examined the turbulent transition in diffusion flames. Now, Professor Hottel was also co-founder with Bernard Lewis and Adrian Arad of the Combustion Institute. And he was elected a member of the National Academy of Science in 1963, the NAE, or National Academy of Engineering, in 1974, and the American Academy of Arts and Sciences. His professional awards include the U.S. Medal of Merit, the Founders Award from the National Academy of Engineering, the Founders Award from AICHE, and the William H. Walker Award from AICHE, among many others. Finally, the purpose of this lecture is to honor Professor Hottel's dedication to both education, science, and energy technologies. He had a broad interest in energy, and his interests are actually very relevant to today. He, with funding from Godfrey Cabot, organized the world's first solar energy utilization research center. His studies led to the choice of a flat plate collector as the most promising device for solar heating, and construction of the first solar heated house, which is shown here, along with 3 others, which provided data for economic assessment of solar space heating and hot water supply. Professor Hottel always advocated for the logical assessment of the economic use of the sun as an energy source. Now, I am really excited to introduce to you Professor Frances Arnold. Now, Professor Arnold is the Linus Pauling Professor of Chemical Engineering, Bioengineering, and Biochemistry at the California Institute of Technology. She pioneered directed evolution, for which she was awarded the Nobel Prize in Chemistry in 2018. And she has used directed protein evolution for applications in alternative energy, chemicals, and medicine. Now, a little bit about Professor Arnold. She began her science and engineering career as a student at Princeton University with a degree in mechanical and aerospace engineering. And inspired by the energy crisis of the late 70s, she worked for the Solar Energy Research Institute, which is now known as NREL, which is a national lab, in Colorado briefly before heading back to graduate school at UC Berkeley for a PhD in chemical engineering with Harvey Blanche. And here she is pictured in her lab at Berkeley, and with recognizable spirit. She actually began to work in the area of biotech during that time frame. And she went on to complete a postdoc at Berkeley and went on to join Caltech in 1986. She pioneered directed evolution and is used to design new enzymes with applications as broad as they are essential, from pharmaceuticals to renewable fuels. Her lab studies its applications in environmentally friendly chemical synthesis and green alternative energy, including the development of highly active enzymes and microorganisms to convert renewable biomasses to fuels and chemicals. Now, Frances has won many awards. Among them, she's received the Charles Stark Draper Prize of the U.S. National Academy of Engineering, the U.S. National Medal of Technology and Innovation. Here she is receiving this medal from Professor Obama, a very significant moment. Brings back good memories. And here we see her, and she has been elected to U.S. National Academies of Science, Medicine, and Engineering. She was also appointed to the Pontifical Academy of Sciences in 2019. It was in 2018 that she received the Nobel Prize in Chemistry, cited for the phage display of peptides and antibodies. Okay, thank you. Antigens. And enzymes. Phage display of peptides and of enzymes.
Directed evolution of enzymes.
Thank you, Frances, for the directed evolution of enzymes.
The guys who won the phage display had.
Thank you, thank you, thank you. She was the 5th woman to receive the award in its 117 years of existence, the first American woman to receive the award, and only the 2nd chemical engineer to ever receive this top prize in its history. Professor Arnold co-founded 3 companies in sustainable chemistry and renewable energy, and serves on several public and private company boards. But she was recently appointed as co-chair of the President's Council of Advisors on Science and Technology, known as PCAST, along with Maria Zuber. And we look forward to her insights on science and its role in policy as she has taken on this critical role. So with that, I'm very happy to turn things over to Frances, who will definitely have it right. Thank you, Frances.
Thank you. Thank you, Paula. And thank you. This is my first in-person talk in years. I love it. I am so tired of yelling at a screen. I can't tell you how excited I am to be here finally. I think we've also tried to have this gathering for a couple of years, and this just makes me feel good to be here. I'm an old-fashioned engineer. I got my degree in chemical engineering in 1985 because I wanted to build new things. I wanted to be an engineer and build new things. And I was so lucky when I came to Berkeley, I took biochemistry for the first time. My first degree was in mechanical and aerospace engineering, and then I learned a little bit of chemistry. But then I took biochemistry, and I completely fell in love with the chemistry of the biological world and the amazing molecular machines that biology has created to do all the chemistry that takes abundant renewable resources and turns it into adapting, moving, sometimes even thinking machines. This is chemistry that we all need to, well, humans can't mimic or master this chemistry, but we need to learn how to use it if we want to build a planet and a society that can, well, feed, clothe, house 10 billion people without destroying the natural world. So I felt that maybe there was an opportunity to become an engineer of this world, to build new proteins. I was so naive. I had no idea how hard this would be because the tools back then, this was really the beginning of the DNA revolution, were so rudimentary that making changes in the DNA sequence that encodes these remarkable molecular machines, where it would take weeks to make a single change, and then you'd go in and you'd use your big brain to design a change, and you'd go in and make it, and then 4 weeks later, you'd maybe, if you were lucky, have a mutant protein, and then you'd test it, and nothing worked, right? Because the people who were doing protein engineering at the time thought that the whole problem was solved, that with their big brains and the few structures they had, they could pinpoint where a mutation would give rise to new properties. And they published only the papers of the experiments that worked. So I went into the field thinking, oh yeah, I could go in just like they did and pinpoint these changes, and I found out that none of my experiments worked because not only did I want to destroy proteins and see how they worked, I wanted to build better ones. I said, okay, I'm gonna make better proteins. So after 3 years of misery, I would say, a lot of you students ask me about, are there low points? Yes, there are low points because none of my experiments worked. I finally figured out some things. But let me just set up this problem. We want to build new proteins by modifying the DNA code. Code determines structure, right? And that recently was pretty much solved by AlphaFold, but structure does not determine function with enzymes. So no one knows how to sit down and design an enzyme. Also, the tools were rudimentary then, but they've changed completely. So let's come to today. You can read any DNA you want. It's almost free. Not quite free, but pretty much free to go in and sequence an entire human genome, maybe less than $1,000 and going down towards 100. That's amazing. You can write any DNA you want, right? You can type off a sequence, email it off to your favorite supplier, and you'll get the physical DNA back in the mail. We had no way to do that 35 years ago. We can edit DNA, right? You can go in and make modifications in a living organism's genome. We couldn't even think of such tools. But what we don't know how to do, and what we still didn't know how to do then and still don't know how to do today, is compose it, right? You can read, write, edit, but you don't know how to compose. What is the sequence that will encode an enzyme that works better than what nature has provided and solves human problems? So that's the problem that I'll talk about today. Now, if you don't know how to do it, what do? You go and look at how it was done elsewhere. And maybe humans might not know how to do it, but nature certainly does. Nature has this remarkable algorithm of mutation and natural selection that gave rise to all the remarkable diversity of the biological world, including all the enzymes, and you. And it's just a simple process of turn the crank of mutation and natural selection. Nobody sat down and designed it. So why not use a process like that? All right, so you have that idea, and it's not a new idea, right? Oh, by the way, so let me put this in context. At the time that I was thinking about doing this in the late 1980s, there were combinatorial phage display methods where people would put a whole lot of randomized sequences on phage and then just sort through billions and millions of them. That's not so easy to do, and I'll give you some numbers in just a minute. All right, here, listen to me, thank you. So this is not a new idea, right? To use evolution to create new biological things has been done by human beings for thousands of years, making everything from corn to carrier pigeons, lab rats, racehorses. We've made things that serve our purposes without knowing even what DNA is, just by choosing who mates with whom and who goes on to parent the next generation. We've made things that are highly non-natural. That poodle, if it got out in my neighborhood in Los Angeles, it would be eaten by the coyotes. It's not even fit. So they would not appear without the intervention of human beings. So we have been breeding biology, making non-natural things for our purposes for a very long time. Now, in the biological world, though, there's some limitations. And in the farmyard, if you're going to breed, or in natural evolution, monkeys go with monkeys, worms go with worms. You're not going to cross these two and get anything even functional, much less better than what you started with. So there are very strict limitations on how you can breed in the organismal level. Even introducing new DNA, you can go out and smoke your cigarettes and probably dial in a few random mutations or take a few more transatlantic flights and get a few random mutations in your genome. But you really don't have much control over that. But all that has changed. Today, of course, because you can write any DNA you want, this is Wolford, it's my laboratory mascot, a photoshopped object, in case you're wondering. We do not cross monkeys and worms, but I could cross the DNA from monkeys and worms. I could have 3 parents. I could have 33 parents. I can dial in any level of random mutations I want. That's both powerful and terrifying in the sense that you don't have any idea what the rules are. We don't have 10,000 years of empirical evidence to tell us how do you breed on a time scale that'll get you tenure, right? Or a PhD thesis, or even solve a real industrial problem on a very short time scale. So we had to figure that out. Great idea, how do you do it? But before I tell you how to do it, let me talk about what to do and why to do it. Any biologists here? All right, I'll insult you first. Biologists, biochemists, have lots of proteins to study, all the proteins that nature has created since the beginning of life. That's your little space, and it's a tiny little green fraction of the universe of possibilities, which is my space. As engineers, we want to create things that don't necessarily exist on the planet, or even may never have existed, but that solve real problems. And I felt that out in this really big space of possibilities that nature has never explored would lie the solution to the energy crisis, the cure to cancer, maybe even the cure to death and taxes. Who knows? And the real question is, in this space of possibilities, how do you get to there in a reasonable amount of time? So you have to believe, and then you have to figure out how to get there. So this is not listening to me. I'm going to move it a little bit. Okay. So it's a really big space. It's a big, 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 big space. If you just take a typical small protein, 300 amino acids long, you got 20 letters in the alphabet, that space of possible sequences is bigger than anything you can comprehend. It's even bigger than the US national debt, which is going to get even bigger. And most of those are meaningless. So let's go back to this context of the guys who are doing phage display, or even RNA evolution. They only had 4 letters in their alphabet, so you can randomize 4 things and have it maybe make a few billion and have a chance of finding something useful. You randomize a protein, you're out of luck, because most of those sequences don't encode anything at all. So here it's like this wonderful short story, and I really recommend this too. If you haven't read it, it's only a few pages long, but it describes better than anything else the despair of the librarians in this library of all possible books, of ever finding a book that had a meaningful sentence, much less a piece of literature. So if you take all the letters of the alphabet and you randomly assort them and you put them in this infinite library, how will you find that true story of your life? So the librarians in Borges' library would throw themselves off the balcony in despair of ever finding that meaningful sentence. So here I am, assistant professor at Caltech, in this library of all possible proteins. It's just a concept, right? And I have to find Moby Dick. All right. So it's not that bad, though. So how do you think about this? It's not phage display. Phage display won the other half of the Nobel Prize. First half was for this. And it's a really different technology because phage display would not work for what we wanted to do. So the numbers grow exponentially as you go and make more than 1 mutation at a time. You've got only a few thousand ways to do that, but you start making many mutations at a time and it quickly goes beyond. And most mutations are deleterious, so you're not going to find anything better. So how does evolution work? How did this problem get solved? Well, it was solved by someone who also wrote a wonderful short story. And this was John Maynard Smith, who wrote the best 1.5-page paper I've ever read in Nature. And he said, well, and this is before sequences of proteins were in databases. You know, imagine that. You couldn't go to a database and see how proteins have evolved. He couldn't go to the database, so he just thought about it. And he said, I think evolution works. What does that tell me about how this process goes? And he said, Imagine a space of all these possible proteins where they're ordered. In Borges's library, they weren't ordered. In this library, however, in his library, they're ordered so that all the single mutants sit next to one another. And that must mean for evolution to work, and most mutations are deleterious, evolution pretty much has to be able to walk one step at a time through functional proteins. And that puts a lower bound on the density of functional proteins around proteins that exist today. And that density is at least one functional protein. So therefore, evolution can just step through these different proteins and create the proteins that exist today. That was a wonderful, powerful idea to me, because I didn't want to just make other functional proteins. You know, evolution just makes things that are not necessarily better or bred for some particular purpose. But for me, it meant that the landscape, the fitness landscape for optimization, should be smooth in some of its dimensions, right? So here, now I'm the breeder of molecules. I decide who goes on to parent the next generation. So I dictate based on my measurements who's fit. And if you plot fitness against all these, multiple sequence dimensions, if that landscape is really rugged, then any mutation will send you into crevasses of non-function and evolution stops. And so Maynard Smith's argument said to me that it has to be smooth in some dimensions, and therefore you can take a really stupid optimization strategy, the simplest possible, random uphill walk, greedy optimization, and perhaps make a better protein. So we just did that in the laboratory, starting with a gift of nature on a silver platter. 4 billion years of natural selection has given you proteins you can scrape from the bottom of your shoe. Those rare functional sequences are everywhere. So if you take that gene for that and you start copying it under error-prone conditions, it's very easy to make lots of mutants, and you control it so that you're only making on order of one mutation. That was really the trick. Put that into E. coli or yeast or whatever, and they'll make mutant proteins. And then the other trick is to measure reliably the benefits of a single mutation. That means you have to do a very careful measurement. If you're going to look through thousands of things and find a twofold increase in catalytic activity, it's not a yes or no, it's a small change. And then you accumulate those over multiple generations, walking uphill. Now, that's what it looks like. You start with an enzyme that does something in nature, you ask it to do something else, and just like people, it's generally unenthusiastic about that. But then you turn the crank and you drive it up the new fitness peak, and that actually works pretty well. All right, I'll just point out for those, a lot of you are doing computational stuff. In the old days, we would throw out all the ones that don't work. Now we just feed them into machine learning algorithms, because now we can get the sequences and the fitnesses of all of those. And so instead of having this simple random uphill walk, so if we look down on the fitness landscape, the random uphill walk will take you to the nearest fitness peak, may not be anywhere near a global optimum, may not even be near a fairly local optimum, but it'll, if it solves the problem, that's good enough. But now today, we don't do things quite the same. We can do a much more intelligent sampling of a local space, identify multiple possible optima, and use machine learning in the process to reach higher peaks and improve this painful experimental process. But I'm not going to talk about that because I want to move on to a different topic. But this process worked, and that's why I got the big prize, because it was easy to do. Any monkey can do it, pretty much. I love things. I mean, that's good engineering, right? If it's really simple. And sorry, if I don't mean to call you a monkey if you're doing it, but it's an algorithmic process, and it's led to all sorts of products that people use, everything from the laundry detergent enzymes to things used in sequencing and making drugs and all that. And all that got me the biggest accolade of my life in 2019 when I got to appear on the set of The Big Bang Theory. Oh my goodness, that was so much fun. I was called up and said, will you come on over and appear on this really silly TV show, and this is MIT, I'm sure you've all seen this. It's about endearing graduate students at Caltech who then grow, after 12 seasons, into professors who are lobbying for Nobel Prizes. So I got to appear on the, they're lobbying for me for the Nobel Prizes. So the good news is I was the only woman ever asked to appear on that show playing herself after 12 seasons. So at least and I have a SAG-AFTRA contract to play myself. The bad news is the show is over, right? So I won't get the chance to do that again, but it was really a lot of fun. And I also, let's see, come on. I'm having trouble getting this thing to work. Okay, I also got invited to a great party in Stockholm. I tell you, if you ever get invited, you should go. And this is Donna Strickland, who was the third woman in history to win the Nobel Prize in Physics. 2018 was a great year because there were 2 separate science prizes, and there we are playing with our chocolate Nobel coins, because you should know that you're not going to get your gold one until you're on the airplane going home, because Nobel laureates are notorious for leaving their coins, their medals lying around, and a couple of them got stolen over the years, so we had to content ourselves with chocolate. All right, so let's go back to science. What did we learn from this? So we made lots of useful molecules, but we also learned some really interesting things about proteins. It wasn't at all clear, it wasn't obvious that you could just turn the crank and move uphill and get anything useful, right? That you would be able to walk sometimes many mutations and make enzymes do crazy, wonderful new things. It wasn't at all clear how quickly that would happen. Would it take 200 mutations like you often see in biochemical systems, or would it take 2 mutations? And what we found is that you could rapidly adapt enzymes by this process. Sometimes only far less than 2% of the sequence would give rise to important changes in function. Now here's the scary fact for you would-be designers. You think you're so smart, you can sit down and compute what amino acid changes would give rise to these properties. We found that many beneficial mutations were lying far from the active site of the enzyme, 20, 30 angstroms away. And when I saw this in the first experiments, I said, ha! You know, I was really happy to see that, because it shows us that biology still has so much for us to learn, right? And so much to teach us. Nobody can even explain these mutations, much less predict them a priori. So that's why 30 years later, people still make better enzymes by this process and not by design. So there's still challenges out there for you to solve. How do you do that? All right, let me switch gears though, because this is what I'm working on now. And that is, how do you actually make enzymes that do something completely novel? Right, I improved enzymes. I improved properties that you could always, you could measure their stability, maybe changing their substrate specificity, very, very useful. But then people were starting to publish papers on novel enzyme design, making enzymes for chemical reactions not even known in the biological world. And I said, that I'd really love to do that. But I was thinking, okay, you have to mold a whole new active site, and that'll take 5 mutations simultaneously, and the probability of finding that is 0. And how do you actually do that? And but then I started, and also, my colleagues in chemistry at Caltech, they say, these enzymes are cute. They pat me on the head, and they said, they're cute, and they have all sorts of nice properties, but their chemistry is really limited. They can't do what I can do, right? And let's be honest, the chemical world is still owned by synthetic chemists. Biology is great, but it has a limited range of chemistry. And so I said, I've got to solve this problem to make enzymes do completely new things and do it better than the chemists can do. So here's the problem. How do you use an inherently conservative process like this, making 1 mutation at a time, which is not going to change the 3-dimensional structure, and get an enzyme that catalyzes a reaction not even known in nature? Well, nature does it all the time. And this is what I should have realized 25 years ago instead of 10 years ago. Should have said, okay, nature does it all the time in response to new opportunities that we humans give the natural world, for example. So if you dump, billions of pounds of a potent herbicide like atrazine on the planet, first it was non-biodegradable. Nobody knew how to eat this stuff. Then a microbe popped up and said, oh, if I take off that chlorine, I'll give myself a rich nitrogen source that gives me an advantage. Of course, that's not what they do. It just happened randomly. But the one that figured out how to do it had a selective advantage in a new food source. And that happened in a real time, 50, 60 years. How did that happen so quickly, right? Well, the bottom line, and this is a really interesting thing to understand about enzymes, is that they're like graduate students. They have multiple capabilities that are not being selected at any one time. Not that they're promiscuous, but they're multi-capable, right? So an enzyme might be selected for a particular biological function, but by virtue of their structure and maybe cofactors, they are capable of doing other things. So graduate student, you're selected for 100 hours a week at low pay, but you could play the piano if you had to, or you could even go do dishes, or you could take on a different career and go over to the Harvard Business School, right? And so my job is to recognize those capabilities and then draw them out through this simple process, right? So Danny Toffick, and I want to just point out, this is a great loss to our community. He died just a few months ago doing what he loved best, climbing. But he did wonderful experiments to show that directed evolution could capture ancient vestiges of activity from ancient activities of enzymes. And he could draw it out by directed evolution. So that just clicked with me, and I said, oh, well then if I can recognize not ancient activities, but proto-activities, things that they could do if you forced them to, or you train them to, maybe we could create new enzymes. So let me tell you a story of that. I started with my favorite enzyme, the cytochrome P450s, which can insert oxygen into unactivated CH bonds at room temperature and pressure, chemistry that humans have not figured out how to do, but an enzyme did a long time ago. And what it does is activate oxygen to make kind of this oxine species. And there's a whole lot of these enzymes around, and they do a lot of really interesting chemistry as a family, presumably evolving through that process like I just described. Some proto-cytochrome P450 organism realized that it could do an epoxidation, and that was useful. And then another one could do a desaturation. And this whole family then diverged through this innovation process. Well, we just looked at this and said, well, if you can transfer an oxine, maybe you could transfer a carbene. Isoelectronic with the iron oxo species, this iron carbenoid, is well known in chemistry. It turns out the biomimetic chemists took porphyrins and used them to catalyze all sorts of carbene transfer reactions. They never could get enzyme-like behavior, but they could get this transfer behavior. And we said, okay, let's go back to the protein and see if we can get them to do that. Right, so we've got inspiration. The chemists got inspiration from biology, we got inspiration from that, and we took it back to biology and said, can you make these carbenes, and then can you transfer those to a second substrate and get them to do a completely unknown kind of chemistry? There's no carbene transferase that you can find in a database. All right, so fast forward a few years, we could do the things that the chemists had done. But then we started saying, okay, we're now got a collection of carbene transferases. That's genetic diversity. Can they start doing things that no chemist can do? So I'm going to tell you a story that no chemist has been able to do, not known in biology. Biology's never known to do it, but a chemist can't do it. It's not even known to be catalyzed by iron. Earth-abundant metal, right? So there was a tiny report of an osmium species that has never been reproduced to make highly strained rings from alkynes. So Kai Chen in my group recently proposed that if you can form the carbene, you can transfer that to an alkyne and make a highly strained cyclopropene. So we're talking 56 kcals per mole, and you use a carbene precursor that's not found in the biological world. You use that to drive the formation of the iron carbonate. If it'll transfer it to the alkyne, you get this cyclopropene. So what does he do? He goes to the refrigerator, tests a whole bunch of our carbene transferases from other projects, and finds a little tiny bit of activity for this cyclopropenation. Then he turns the crank of evolution and makes the most efficient cyclopropenase that anyone has ever made, good enough to get into a snooty journal. But he didn't stop there. He said, oh, cool, I've got a double bond on the cyclopropene. What if I can get the enzyme to hang on to that highly reactive intermediate and do it again and make a bicyclobutane? Wow, that's pretty cool, because now we're talking, rocket fuel binder kind of stuff here, 70 kcals per mole, and no one has ever made a single stereoisomer of these things. I'm not sure anybody wants a single stereoisomer of these things, but this is something no chemist would have any clue how to do. Yet, Kai was able to find a tiny bit of this activity in one of his cyclopropenases and evolved the second step to make gram quantities. And we're now supplying these to the Army for energetics. And this is not a trivial catalysis thing, right? Because you're starting with a cofactor that has no measurable activity. So it's somehow activated by the protein. You're asking it to go through two distinct orientations in the transition states. So your catalyst has to accept these two different orientations and keep the perfect stereoselectivity, and you've got an unstable intermediate. I defy you to design a catalyst that could do that. Yet evolution, just turn the crank and you could get that. So he was able to make, as I say, gram quantities. We could get crystal structures, show the particular orientation. And that was a lot of fun. And as I say, we're collaborating with the Army. So back to my Caltech colleagues' criticisms that the scope of chemistry in the biological world is limited. To some extent, they're really right? If you look at CX bonds known in biology, carbon goes with hydrogen, lots of hydrogen. Carbon goes with nitrogen, oxygen, some sulfur, a few halogens, a few metals. But there's whole swaths of the periodic chart where at least we don't know of natural products that contain those. Biology hasn't made those. Yet humans do it all the time. Humans love the periodic chart and love making crazy molecules that biology has never explored, or at least we haven't found them yet. And one great example is carbon-silicon bonds, organosilicon, huge industry. There are 50 products in this room, in your hair and your earbuds, that have carbon-silicon bonds. It's a mega industry, Dow Chemical, for example, all done with nasty human chemistry. Platinum catalysts lead to tremendous environmental degradation. Wouldn't it be cool if you could genetically encode the formation of carbon-silicon bonds? Well, you can do that. In fact, all you have to do is ask. We went to the databases. I didn't go to Iceland, but we found a very small protein from a hot, salty pool that was found in Iceland. And it turns out it's a cytochrome C will catalyze this reaction. So it's only 124 amino acids. It's just this little rock of a protein. You can boil this thing. It's so stable, you can give it to a chemist. You don't even have to tell them it's a protein, right? You just put it in a bottle and say, here's my magic, silicon carbon bond forming powder. And it has no catalytic activity that was known, electron transfer, yet if you take a silane and the carbene precursor, it'll make a new carbon-silicon bond. Now, 40 turnovers, that's as good for asymmetric carbon-silicon bond formation, asymmetric carbene transfer to a silane that was ever reported by a human being. That's as good And it's just sitting out there in nature on the bottom of your shoe, waiting for you to ask whether it could do that. And it turns out the answer is yes. And when I talked about this work, I had a cytochrome C expert sitting where you were sitting. And he jumps up and he said, but, Francis, don't you know that the active site is no active site, right? That the whole coordination sphere of iron is occupied. You've got a methionine above and a histidine below, and the porphyrin occupies all the equatorial sites. So how does this happen? How can it happen? He didn't believe me. And I said, well, all right. So if you look at the structure and you roll your little ball, right, your little computer ball over the active site, you come up, you're right, you come up with a big fat 0. But you know what? Nature doesn't care about your calculations. Because this protein catalyzes the reaction. And this, obviously, our puny minds do not comprehend the complexity of these dynamic molecules. And not only does it catalyze the reaction, but it evolves. Right? So if you start directing mutations to residues in that active site, over just 3 generations of random mutations in there, you can come up with a mutant that does it 50 times better than the best human chemist. Right? So it's very easy to evolve this, and you can make lots and lots of carbon-silicon bonds. In fact, you can do it inside of living bacteria. These precursors will float right in, and the products will float right out, and you can do it with remarkable selectivity. And in fact, so when this paper was published, this is a little story in scientific publication. The paper, we had to do so much work to get it published in Science. The SI alone is 130 pages because we made 20 new organosilicons that nobody had ever characterized before, and they insisted we characterize every single one, so NMRs and all that. So all that stuff is in there, and it's really painful. Nobody cares about these molecules anyway. The whole point was that you could program bacteria to form carbon-silicon bonds. So the paper is really boring, but the science writer says, oh, that's so cool. You know, I've got to write about it. And this was before the paper was published. And I said, well, I can't, you got to zip your lip and not talk to reporters. Otherwise, you get in trouble for publication by press release. So I said, no, you got to write based on what I said in the talk. So he says, he publishes this artwork. We're taking a step towards silicon-based life. And he puts Dr. Spock and Captain Kirk with a Horta episode where they're looking for life in rocks, So no one read my paper. Everyone read this. And the news went all around the world that we were doing things like silicon-based life. And so, popular science, at least they know something about chemistry. And they said, I don't think so. We never said anything about, there's nothing about silicon-based life in my paper. But I think, this is going to happen, but Astrobiology magazine, they really liked the idea, so all 17 readers of Astrobiology magazine clicked on my article, making it the highest read article for the year. And then, in Evolution News, this is the creationist community, they said, this has got to be intelligent design, And I said, yes, I am the intelligent designer. Here I am. And if you ever want to roll on the floor laughing, get Twitter to review your paper. So we had a lot of fun with the Twitterverse response. I still love, if you want to know what the world will think of your paper, put it out on Twitter. All right. But what's cool is that with this powerful process, we not only added silicon to the periodic table of the biological world, you now can make organosilicons genetically encoded, fully genetically encoded in living systems, and boron. Very similar story for boron. So now you can make organoborons and organosilicons. So now you metabolic engineers start thinking, organosilicons, silicon-based life. All right. So I could tell you a whole other story. This is not just a one-off thing. Carbines are really cool. How about nitrines? Nitrines. So chemists know all about nitrines. You transfer a nitrine, you get a new carbon-nitrogen bond. And that's chemistry that was completely unknown in the biological world until we showed it was possible. And then some scientists in Japan actually found a nitrine transferase in nature. And they hadn't understood the mechanism, but it's transferring nitrines. So we did a lot of work on these nitrines, and I'll just show you the very latest one that we published. No chemist does a good job at this. If you take a hydrocarbon and you try to just plug on a free amino functionality, that is not easy to do. But and it's not known in nature. Nature makes amines through a very different process. And in fact, people who do biocatalysis, right, you might do an oxygenation and then an amino transferase. Nobody knows how to do a direct plug-in of an amino functionality, but these nitrine transferases made in the laboratory can do that. We've shown it with benzylic and allylic, and next year it's going to be unactivated CH bonds. Enzymes can learn how to do this chemistry if given the sufficient push by the breeder of molecules. So this is an exciting area, and I think we'll see a lot of work from the nitrine. Basically, what I want to summarize now, I've tried to show that this powerful process that has given rise to all of life and all the chemistry of life has not stopped. And you can use it to move, nature will use it to move into the future, but you can do it too. So if you like to think about microbes as chemical factories, now you can start thinking about how would I marry my favorite chemical transformation and encode it in DNA. So that, for example, we might be able to use abundant renewable resources to make pretty much everything that we want to be able to use. And I also want to point out, you know, we all talk about the Internet of Things and how all these objects are talking to each other. That can't hold a candle to the Internet of living things that has been problem solving by crowdsourcing for nearly 4 billion years. Exchanging DNA back and forth, adapting, evolving, creative, innovating. And if we learn how to use this process, we might even be able to adapt and evolve and innovate in tandem with our beautiful planet. So I will thank all the young people who did all this work. I just go around the world getting credit for it. That's the nature of the job, folks. So but it's a wonderful thing to be able to work with people because they come in with all these fabulous ideas. Nothing that I talked about here, none of the specific projects that I talked about in the second half of the talk were my idea. They were the ideas of chemists who now can look at proteins in a whole new way. Not for what they do, but what they can do. And say, okay, if I'm a good trainer, what is the proto-capability of this student, of this enzyme, of this protein that I can draw out through this process of evolution? So thank you.
Thank you, Francis, for that incredible lecture. We have learned so much from you, and we are so happy that you were able to come and join us. I would like to present you with the silver bowl, which is the traditional gift for the Hoyt Hottel lecture.
Ooh, I like that. Thank you.
Engraved your Hoyt Hottel lecture.
Thank you.
And we will ship this to you. And
no, I'm going to put it in my bag. It doesn't weigh very much. That's good.
All right. Excellent.
It might get damaged in shipping, and plus this is useful. Thank you.
It's useful. Absolutely.
Thank you.
And we will open up to questions. I'm going to invite people who have questions to come to the microphone. If there's difficulty, we will start passing it. But because we're on live stream, we would like to capture as much as we can on the mic. So if anyone would like to come on either side to the microphone. If you're way too distant.
Oh, don't be shy.
Raise your hand.
Francis, that does the ability to climb up the property depend on what kind of a phenotype you are looking at? For example, hydrophobicity or a binding ability is different than activity in an enzyme. Do you expect all of them to follow the same trend?
Well, most things, so affinity maturation of antibodies, for example, is an uphill climb. A lot of things can evolve that way. The real trick is not to ask too much, to jump too high in each generation. And sometimes you have to take a few circuitous routes, right? But I mean, I have run into a few things that just won't evolve, and it's not clear why that's happening. So maybe not everything will do it, but enough to get me the Nobel Prize, I guess. It's just that so many people use this process for so many different kinds of problems that it must be a universal property that can be exploited. Oh, there we go. I'll take this off. Yeah.
It would be wonderful to have some students ask questions if they'd like.
Hello? Okay. So what advice do you have for like a growing scientist today's day and age?
I don't like to give specific advice. Every path is different. Every person's path is different. I took a very random and circuitous path, but all those experiences, they really did make me who I am. I tried a lot of things that didn't work, and I, learned things that I didn't think I would use, and then was able to recombine later on. So my only advice is collect knowledge like it's money in the bank. And collect that knowledge and be flexible, and maybe you can recombine it.
Sure, there's a question here, and then would you like to come forward with this question?
Yes. So as we were taught in textbooks, biology does not do things that it does not need for itself. So I always imagine that if biology did not end up like making carbenes or nitrines within itself, then it just didn't need them. For the work that you have been doing, you have implicated it in systems outside of biology, using biology. Do you envision that these systems will be useful for biology itself as well?
So that's a really interesting question. You're absolutely right. I've just used biology as a bag in which to make enzymes. And the carbenes were driven, the formation of carbenes were driven by non-biological diazoacetate. There's not large reservoirs of diazoacetate or diazos in general. However, the nitrines, what we're finding is there are reservoirs of those. And my guess is somebody will discover a carbene transferase at some point, right? I think biology can do, obviously, biology can do anything I've done because we're just doing very local explorations. Biology can do it. She probably, just doesn't care about some of the things that we make. Frankly, nobody else cares about some of the things we make. But I think that there's a lot of more exploration for us to do, and we'll be surprised. So I hope that somebody will publish the first natural carbene transferase.
Hi, thanks for a great talk. So I like to think about protein design and how it works when you have amino acids that are kind of weird, like different side chains, but also like, beta amino acids, D amino acids, like all that stuff. I was wondering, when you think about protein design with those kinds of building blocks, do you think of them the same way you think of other amino acids? Or do you have to think about it in kind of a different way because they're not really part of nature?
Yeah, so there's a lot of interest in expanding the alphabet and expanding the chemistry, right? So expanding the alphabet only makes sense if you expand the capabilities beyond what you think you could get in that universe of possibilities with the 20 natural amino acids. And there's some decent arguments for specific functions, you can get that. But that's very hypothesis driven. And I'm sure you can show examples of that. I have stayed away from all that. And the reason is I really want to explore the universe that nature has given us. And what I've found is that you can do all sorts of surprising things. The whole world is publishing these artificial metalloenzymes, for example, using non-natural amino acids. Our proteins are so much better because they evolve, right? It's very hard to do directed evolution on those technically challenging systems, but we can do directed evolution really quickly and make up for all the bad human design. So until you get a technology that allows you to use evolution, there's not a whole lot of advantage to expanding the alphabet for enzymes, in my opinion.
Okay, cool. Thank you so much.
Yeah.
So I have a pretty common question. Oftentimes we learn a lot from the failures in science, not just the successes. So I was curious if you could share an instance of where you tried to evolve an enzyme for a certain function and it didn't work, and why it didn't work.
Well, the most interesting time we tried to evolve and it didn't work, I learned something super important. So I had a student who kept cranking on a cytochrome P450 and said, Francis, I just can't find any more beneficial mutations. And it was a terrible enzyme. I said, oh, come on, there's going to be a lot of room for improvement. So I was sure that this student was just not doing the experiment right. And then, okay, so I had somebody else come in and try it. And that student realized that what was going on is that the protein became so unstable, had become so unstable that it just wasn't accepting any more mutations, right? You would make random mutations in it, and because most mutations are deleterious, they were all just unfolding, and it didn't happen until you made the protein more stable. And any optimization scientist knows this, but we had to relearn it. It wasn't until you made it more stable that you could go in the other direction of making it more active. And that led, so that was Jesse Bloom, and led to one of the most highly cited papers that we ever had, which was Stability Promotes Evolvability. And it's a very simple idea that if you don't have a reservoir for accepting destabilizing mutations, you won't be able to explore those beneficial activating mutations. Yeah. So you got to take your lemons and make lemonade and kind of try to understand why it didn't work, and maybe you can learn something super interesting.
Thank you.
Hi, Professor Arnold. My name is Michelle. I was just curious about your thoughts on going back in time in terms of the enzymes and going back to, I guess, the state where it is not as specialized to do its function and re-evolving for another function from there.
I don't see any reason to do that. And I'll be really frank. I don't think ancestral reconstruction, because there's a lot of ambiguities about exactly what the sequence is, gives you a good idea of what the promiscuous activities were. Because I know by experiment that if you do a few random mutations, you get promiscuous activities. So it could be just a failure in the ancestral reconstruction that gives you those features. So I would rather just take the enzymes, throw in a few random mutations, make them more promiscuous, right? Accept a different range of substrates and move forward from there.
Okay, thank you.
Awesome. So first of all, just thank you for the lovely talk. I really, really enjoyed listening to just about your chemistry, very unique. My question is, how do you envision that this kind of work and these unique reactions can be implemented onto the industrial scale in order to address major climate change issues and other kind of global problems?
Oh, there's a lot of possibilities, and one of the projects that I'm really excited about is we're making enzymes to degrade organosilicon. We can make organosilicons. How about degrading them? Those are considered non-biodegradable, and they're accumulating everywhere, right? So if you can make enzymes that can degrade these things, we already showed last year that you could hydroxylated silicon. Nobody knew that, right? So there's possibilities for doing things that would be really useful.
Absolutely. Thank you.
Yeah.
Thank you, and thank you, Francis. It's a wonderful lecture and opportunity to hear your thoughts.
Thank you.
Thank you.
Thank you, everyone.
Okay.